Welcome back to Biology Chemistry. This is Unit 1, Lesson 2 on the scientific method, video number 2. So we've gone over the first two steps of the scientific method, identifying the problem and writing a hypothesis. Now we're going to move on to the third step, which is designing an experiment. When we design an experiment, it must test the hypothesis. So we made that if-then statement. Now we have to design an experiment to test that statement. So whenever we design an experiment, things that we need to include are a list of materials that are needed. So providing that list, because one of the big things is we have to make sure that we can repeat this experiment. So if somebody else wants to try it, they need to know what they need to do the experiment. And the other thing is we have to design a procedure. So a procedure is just step-by-step explanations on how to conduct the experiment. So it's almost like a recipe that you would follow. Do this using these materials, do this, do this, do this, collect this data, and then follow that procedure. So one thing that we have to talk about, though, when we design this experiment are variables. These are factors that change in an experiment. Now, in a proper experiment, and this is one of the most important statements that we can make here, is only one variable can be changed in a proper scientific experiment. Because we want everything else to remain the same, so we know if we change that one thing, that is causing whatever effect that we see. So whenever we talk about variables, we are going to have an independent and a dependent variable, one that is changed and one that's resulting. So you'll see that I have these words here, manipulated and responding. So if you can associate independent with manipulated and dependent with responding, that can help explain it. Because an independent variable is a variable that is tested and changed by the experimenter. So the scientist who's running the experiment. So this is the one that is changed. Dependent is responding. This is the variable that is measured and changes as a result of the independent variable. So when we go through and look at an example, we'll talk about what is independent or manipulated and what is dependent or responding. So if we go back and talk a little bit about one of our hypotheses, well, that moldy bread one, if I eat moldy bread, then I will get sick. Well, what am I manipulating? Well, I am eating the moldy bread. So the moldy bread is the independent variable. What am I seeing? What is responding? Well, if I get sick or not. So the thing that's changed is manipulated, and what you're looking for and what is measured as a result of that change is the responding variable. All the other things have to be controlled. So these are variables that are kept constant or the same throughout the experiment. So if I was doing a controlled experiment, this is an experiment in which one variable changes and all others are kept the same. This is the goal of any experiment, is to be controlled. So if I was looking at something maybe on the temperature of my classroom and grades, so everything else would have to stay the same except for the temperature of the classroom. So I would have to give the same test to one group that another group has. It had to have the same number of people. It would have to have the same number of boys and the same number of girls. Everything except the temperature in the room, the time of day would have to stay the same. The amount of sunlight coming into the room, whether lights are on or off, all of that stuff has to be the same except for the temperature in the room changes. That way, all that other stuff has no effect because it's the same.